Hi doctor friends, welcome to my YouTube channel PG Doctor INDRANJU. Today's topic is about the GIT physiology. Let's start with GIT physiology. This is very important topic for the first year student and for the which student preparing for the NEET PG, AIIMS PG, FMGE and all types of medical PG. This is the GIT whole physiology. So start with the GIT physiology. Alimentary canal. Alimentary canal, the cross section of the alimentary canal as shown in the diagram. This is the diagrammatic representation of the cross section of the alimentary canal. Here I am showing the outermost layer is the serosa. The serosa is also covered with the mesentery, and so the mesentery and serosa, between that layer it is the connective tissue and the epithelial lining. Start with outer to inner layer. The outermost layer is the serosa, then after muscularis mucosa. The muscularis mucosa, the outer lining is outer longitudinal layer, then after inner circular layer. The pattern is showing the circular in nature. Here circular nature, due to cross section it is a vertically represented longitudinally arrangement, and in the cross section it will appear as circular. After that the inner circular layer, in the cross section it is like circular pattern. So outer longitudinal, inner circular, then after layer is submucosa, after that mucosa, and inner most part is the lumen. The mucosa have lining epithelium. If start with the stomach, it is columnar epithelium, and in the small intestine, due to absorptive property of nutrient and electrolyte, so that is presence of villi, so that make it the surface area larger, so that absorption takes place easier. Then after here the innervation of the GIT system from esophagus to anus. The here type of the nerve supply is enteric nervous system, enteric nerve supply or enteric nervous system. This is two types. First is orbital plexus, also known as mentric plexus, and Meissner plexus or submucosal plexus. Due to presence of Meissner plexus in the submucosa, so also known as submucosal plexus, and the property of the Meissner plexus is for the GIT secretion and the local blood flow. Here the submucosal plexus is present in the submucosal layer near the mucosa and have the gland, so that it secrete the mucus secretion, and due to secretory property and local blood flow in that. Next is the orbital plexus or mentric plexus. It is situated between the longitudinal and the circular muscles layer, and property of that, due to presence of in the circular and longitudinal layer, due to present in the muscles, so have property to contract and relaxation. So for the peristaltic movement and presents in muscles, so it propagate the food content or food, digested food, forward manner. Next is second nerve supply is the extrinsic nerve supply. Extrinsic nerve supply, here the nerve supply is the that for the only modify the activity of enteric nervous system. The enteric nervous system is self-regulating, but extrinsic nerve supply is only for modification. And extrinsic nerve supply is the ANS, that means parasympathetic and sympathetic. It connected to the intrinsic nervous system or nerve supply. Then after the GIT secretion. GIT secretion is the first salivary secretion. Salivary secretion, it is daily output of the salivary secretion is 1 to 1.5 liter per day, in which 250 to 400 ml from the parotid gland and 700 to 1 liter submandibular gland and 50 to 100 ml from sublingual gland. Here the parotid gland is purely serous, while that of submandibular gland is the mixed gland, and sublingual is the more mucus than serous fluid. And the pH of the salivary secretion is 6.5 to 7 and is always hypotonic to the plasma. And salivary amylase or ptyalin is secretion of the salivary gland. And lingual lipase, also known as Abner's gland, which are situated in the dorsum of the tongue. And the secretion of the salivary gland is the immunoglobulin G and A, mostly A. All the secretion from the body contains the IgA and also some IgG. Next is the gastric secretion. The gastric secretion 1.5 liter per day or 105 ml per hour. This is due to asked in the NEET PG that the gastric amount of gastric secretion, it is 105 ml per hour. It was given. The major secretion of the gastric secretion is the body and fundus of the stomach, which are mucous gland. The gastric secretion in the individual manner, I am showing you the parietal or occipital cell. This is many times repeated question, so you have to make such type of notes. The parietal or occipital cell secrete HCl and intrinsic factor, while that of chip cell, peptic cell or zymogens all are same, which secrete pepsinogen. Here the pepsinogen, gen indicates the zymogens or inactive enzyme. When the HCl is present, the pepsinogen is converted into pepsin for the active enzyme group and functional enzyme. Next is the G cell. G cell, already know the word G, so it is gastrin. Next is the delta cell. Delta cell secrete somatostatin, while that enterochromatin-like cell which secrete histamine, and gastric mucous cell which secrete the bicarbonate ion. Next is duodenum. Duodenum, the Brunner's gland situated in the duodenum. Many times asked that Brunner's gland situated, so it is in the duodenum, and it is for the secretion of the thick alkaline mucus and bicarbonate ion. The duodenum secretion by the two sources. First, pancreatic juice. The pancreatic juice secretion is 1.5 to 2 liter per day. The mainly here the description about the exocrine part. Endocrine part described in the endocrine. The pancreatic juice secretion is isotonic to plasma, while that of the salivary secretion is the hypotonic. Here pancreatic secretion is the isotonic to the plasma. So the pH, maximum pH of the pancreatic juice is the 8. So bicarbonate, due to pH is 8, the bicarbonate is in the maximum concentration. The pancreatic juice have all types of enzyme so that it can digest all types of the food or all components of the food. Then the secretion of the pancreatic juice is the secretin. Secretion from the pancreatic juice is the trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase A and B, procolipase, pancreatic lipase, and alpha amylase, bile salt, and the ribonuclease. These secretion are in the inactive state after favorable environment like that of the trypsin and CCK. This is converted into active form, and this active form acts on the respective substrate. Next is the bile juice. Bile juice also called as the bile, and the pH of the bile juice is 7.8, and the secretion is 500 ml per day or 20.8 ml per hour by the liver. And the bile juice which are secreted in the duodenum get absorbed in the terminal ileum. If we talk about the bile component contains the bile salt, bile pigment, lipid, and some electrolytes. The bile salt have the taurocolate, glycocolate. Bile pigment contains bilirubin and biliverdin, and the lipid components includes the cholesterol, lecithin, fatty acid, and triglyceride. And electrolytes includes sodium, chloride, bicarbonate, all types, maximum types of the electrolyte. We are discussing about main things which are related with your exam. The bile salt have property and function to emulsify the fat and micelle formation, which are property of the cholecystic function. Next secretion is the small intestinal secretion. These are also isotonic to the plasma and the pH 7.5 to 8. And these are intestinal gland and epithelial cell secretion. The intestinal gland secretion, this includes the crypts of liver cone, which secretes a secretion known as succus entericus, and the Brunner's gland, which are only present in the duodenum, have mucus secretion, and the goblet cell, whole GIT system, whole small intestine, which are also mucus in the nature. The epithelial cell are the brass border columnar, and secretion of the epithelial cell of the small intestine is the disaccharides. Disaccharides include sucrase, maltase, lactase, telomerase, and the isomaltase, also alpha limit dextrin, peptidase, intestinal lipase, enterokinase. After that, our theoretical discussion on the concentration. These are also important. The concentration of the potassium concentration maximum in the colon, after that saliva, then gastric and a small intestine secretion, bile and pancreas have equal secretion of the potassium secretion. It is concentration. So question arising that the maximum concentration of the potassium present in the colon. So if the diarrhea takes place, there is loss of potassium. And the concentration of the sodium concentration is maximum in the bile, pancreas and the small intestine. It is just reverse of such. After that, GI, colon and the saliva. Here the just reverse in such way. So the sodium concentration is maximum in the saliva. Then after, minimum concentration in the saliva and maximum concentration in the bile, pancreas and small intestine. The concentration of the chloride and maximum concentration of the chloride in the small intestine, then bile, then gastric, after that the pancreas, after that colon, then saliva. The one important things that the chloride ion activate both salivary and the pancreatic amylase. Next is the pro-enzyme or zymogen or inactive enzyme. This is actually 3D structure of the protein, and it is when activated gives the active form. And here for the activation, the small length of the protein or amino acid are cleaved off from the one end of the pro-enzyme, which get converted into the active form or enzyme. And here the discussing topic is the GIT hormones. GIT hormones belongs to two categories mainly, gastric family and the secretin family. Gastrin family includes gastrin and CCK, while that of the secretin family, this includes the secretin, glucagon, VIP and GIP. The start with individual hormone, individual enzyme, the gastrin. These topics are very important for you. Start with the gastrin. The gastrin secretion by the G cell, a G cell of the gastric mucosa in the antrum of the stomach and the first part of the duodenum. So gastrin secretion from the antrum and antrum of the stomach and the first part of the duodenum, and also from the pituitary gland, fetal pancreatic islete cell, and brain and nerves. The many forms of the gastrin are present. Main form of the gastrin are G17 and gastric, which are present in main secretion of the gastric acid. And topic is the substance that aggravate the gastrin secretion. Here the products of the protein digestion. Product of the protein digestion includes the tryptophan, phenylalanine, and gastric distension, which also secrete gastrin, and vagal stimulation. Vagal stimulation, which includes mainly autonomic nervous system, and autonomic nervous system, the parasympathetic postganglionic nerve supply is main for the secretion of the gastrin, and it releases gastrin releasing polypeptide instead of acetylcholine. Here the neurotransmitter is PP. The trick or mnemonics for the increased gastric secretion is the good peptide fast to digest. Good peptide. Good peptide. Good includes the G and D. This includes the gastric distension. Peptide includes protein, which are phenylalanine and the tryptophan. And vast that means the vagal stimulation. And to digest for the protein digested amino acid. Next is the substance that decrease gastrin secretion. This includes the acid in the stomach, somatostatin, the already named as statin, that means static, which static the gastrin secretion. Secretin, that means it secrete in the small intestine while inhibit gastric secretion. Gastric inhibitory peptide, VIP, CCK, cholecystokinin, calcitonin, and glucagon. These all are repeated question, so you have to remember by mnemonics, by your own mnemonics or these mnemonics. Mnemonics is that the less gas in the stomach create some secretion of the acid with VG inhibition. Less gas, that means inhibit or decrease gastrin secretion. And stomach for remembering, create includes CCK, some includes somatostatin, secretion includes secretin of the acid in the stomach with, that means C, with is written as C to the bar, and C for the calcitonin, and VG, that means BIP and GIP, inhibition, that means the inhibit. Next is the action of the gastrin. The action of the gastrin is for the gastric acid secretion, pepsin secretion, increase gastric motility, and insulin and glucagon secretion. Next is cholecystokinin. Cholecystokinin have the property is that it is secreted by eye cell of the intestinal mucosa of the small intestine, and substance that stimulate CCK secretion is the product of the protein digestion peptide and amino acid contact with mucosa. Here the long chain fatty acid more than 10 carbon in the duodenum also secrete the cholecystokinin. The function of the cholecystokinin. Here a lot of function of the cholecystokinin. So you have to remember such type. The GB contraction and relaxation of the sphincter of the body. This is the function. The pancreatic juice secretion and increase the action of the secretin, decrease gastric emptying and increase the contraction of the pyloric sphincter, increase motility of small intestine and increase the secretion of the enterokinase and increase insulin and glucagon secretion and relax less lower esophageal sphincter. Next is secretin. Secretin is secreted by ash cell in the duodenum and the jejunum. The substance that stimulate secretin secretion is the acid in the duodenum, most important, and the content of the product of the protein digestion in the duodenum and the jejunum. The function of the secretin is the increased secretion of the pancreatic fluid. This includes water, bicarbonate, and the lase enzyme. It stimulates action of the CCK and stimulate bile secretion and decrease acid secretion, increase tone of the pyloric sphincter, so decrease gastric empty. Here I am showing the mechanism of gastric acid secretion. This is the stomach and the part of the body. Here the diagrammatic representation of the lining of the stomach so that you make it clear. Here this is the vessels which are outer in structure, and in that beneath that there is the parietal cell. Parietal cell, this is the only diagrammatic representation so that you can easily understand it. And here the parietal cell are situated in this part, and outer part have the vessels, and this is lumen part, that means this lumen part. And here something important is here the parietal cell contains water and CO2 is combined in presence of carbonic anhydrase and it form H plus ion or bicarbonate ion. This bicarbonate ion is transported to the interstitial fluid and then to the vessel due to alkaline tide or due to washing out of the bicarbonate. This is active channel, so it requires energy, and in the vessel to interstitial fluid there is due to transportation against exchange of the chloride, and this H plus is transported to the lumen, and in the lumen there is potassium ion present, which are transported to the parietal cell. This exchange is guarded by ATPase channel, and the chloride ion, which are transported from the interstitial fluid to the parietal cell, it is also transported to the lumen, and this H plus and Cl minus combine to form HCl, hydrochloric acid. This basolateral membrane and the vessels, the gap between them is known as the interstitial fluid. The substance that stimulate gastric acid secretion is the histamine, H2 receptor, and the due to dominant role, and acetylcholine, M3 receptor, vagal stimulation, also stimulate gastric acid secretion and gastrin. Here it is gastric acid secretion, not gastrin secretion. I have talked about the gastrin secretion. This is the gastric acid secretion, which are stimulated by histamine, acetylcholine, and the gastrin. Next substance that inhibit gastric acid secretion is the somatostatin, which directly to the parietal cell. Secretin, CCK, GIP, VIP. This is indirect secretion. This secretion, first of all, activate somatostatin, and somatostatin directly inhibit to the parietal cell for the gastric acid secretion inhibition, and also the gastric acid secretion inhibited by the PGE2 and acid and low pH, that means it is based, which inhibit the gastric acid secretion, and vagotomy. The vagotomized patient have less gastric acid secretion, which are suffered from the peptic ulcer disease. Then start with the phase of gastric acid secretion. This is divided into three phases: cephalic phase, gastric phase, and intestinal phase. The cephalic phase includes no content of food taken, only the stimulus is through the vagus nerve, and it is stimulation of the parasympathetic system by sight, smell, thought, which stimulate the secretion of the gastric acid. It is 20 to 30% of the gastric acid secretion. Then after the gastric phase, this includes the hormonal stimulation due to gastrin release, and the stretch stimulation. That means the food content which are taken and which are goes to a stomach, and distension of the stomach have a stretch receptor which are stimulated, and here the vasovagal reflex is stimulated, and local intragastric reflex are stimulated, which are responsible for the gastric acid secretion. Next is the intestinal phase. That means the food when the food enter to the small intestine from stomach. Here the food entered in the duodenum, gastrin, but the major role is the inhibition. This includes the hormonal, that means the stimulates secretion of the when food enter in the intestine, then a stimulation of the secretion of the secretin, CCK, VIP, GIP, somatostatin. This inhibit gastric secretion and gastric motility also. Next is the neuronal mechanism. Neuronal mechanism is include the enterogastric reflex. Next is the topic is the gastric acid output. Gastric acid output, this includes the basal acid output, also called as the BAO, the rate of acid secretion without any other stimulus. It is called the basal acid output. This use in dwelling catheter from 9 p.m. to 9 a.m. and normal it is less than 10 millimole per hour. This is calculated by the 9 p.m. to 9 a.m. which are indwelling catheter are inserted. Then after it is calculated, it is basal acid output. No stimulus are given for the secretion. Then topic is the maximum acid output. Maximum acid output, it includes use of a stimulus like pentagastrin, here the pentagastrin, or histamine. It includes when it started stimulation, then taken every 15th minute for 1 hour, that means 4 times, and normally it is less than 50 millimole per hour when maximum stimulation takes place by the pentagastrin or histamine. The all mixed food and the gastric secretion in the duodenum is the called chyme. Here the maximum acid output are due to risky in case of peptic ulcer diseases and like which tumor that have property to increase the HCl secretion, so this is risky in that condition. Here the bile juice, start with bile juice. The bile juice, the secretion is 500 ml of the bile per day. I already told the most of the bile reabsorbed in the terminal ileum by the enterohepatic circulation. The composition already told, the bile salt, bile pigment, lipid and electrolyte. Bile salt include taurocolate and glycocolate, and bile pigment include bilirubin and biliverdin. The function of the bile for the fat emulsification, micelle formation have cholecystic function. And the topic is about the primary bile acid which are synthesized from the cholesterol in the liver. This is cholic acid and chenodeoxycholic acid, and the secondary bile acid which are the primary bile acid in the intestine is converted to deoxycholic acid and lithocholic acid. These are secondary bile acid due to converted from the primary bile acid. Then after the bile salt have property for emulsification, micelle formation. Due to detergent action of the bile salt, it lower the surface tension. The regulation of the secretion, the cholecystic property, cholecystic and cholegob. The cholecystic, that means substance that is increase the hepatic bile secretion, that means bile salt, and the secretin. These are most important. And the cholegob, substance that cause gallbladder contraction, which are CCK, most important, and also fatty acid and amino acid. Next, our discussing topic is the digestion and the absorption. Start with carbohydrate digestion. Our carbohydrate foods includes mainly starch. So first, digestion. Digestion of the carbohydrate starch is started in the mouth, then in the intestine, that means in the duodenum, while absorption of the hexose, when starch is break down to the simplest sugar, that means hexose mainly, and the pentose in the proximal part of the jejunum. Here the major absorption takes place. Then discussing topic is the what is the process of the digestion. So start with mouth. The start here in the mouth, the salivary amylase and ptyalin are present. Main substrate is the starch. I have already told chloride activate both salivary and pancreatic amylase. Here the salivary amylase or ptyalin break starch into maltose, maltotriose, alpha-limited dextrin. This includes 8 glucose molecules which are binds by alpha-1,6 linkase. Here the mouth, I have told the digestion of a starch in the mouth. Then after duodenum, stomach have no digestive enzyme for the digestion of the carbohydrate, so it is spared. The duodenum digestion starts with pancreatic amylase and intestinal secretion. Pancreatic amylase, both amylase, only this amylase break the internal alpha-1,4 linkase, not terminal and alpha-1,6 linkase. The amylase have property to break only internal alpha-1,4 linkase and have no property and not able to break the alpha 1,6 linkage and the terminal alpha 1,4 linkage. The discussing about the intestinal secretion. Here the oligosaccharides. Oligosaccharides includes maltase, lactase, sucrase. It is the maltase acts on maltose which break to the glucose and glucose molecules. Lactose is the carbohydrate substrate. Here the lactase enzyme break to the glucose and galactose. Sucrose get breakdown to fructose and glucose by sucrase enzyme. So you have to also remember the maltose, the breakdown product of the maltose is the glucose molecule. Lactose includes glucose and galactose, and sucrose includes fructose and the galactose. Next is the only enzyme that break alpha 1,6 linkage is the alpha limit dextrinase or isomaltase. Okay, alpha limit dextrin and isomaltase break down into the glucose. So you have to remember the alpha 1,6 linkage, the alpha 1 limit dextrinase and isomaltase have property to break alpha 1,6 linkage, and the what will be the absorption. So the glucose and galactose, this is important topic. The glucose and galactose is absorbed from the lumen of intestine, that means mainly jejunum, by the secondary active transport with sodium and co-transporter. Here the carrier protein is the SGLT1. Okay, and here first the digested carbohydrate is glucose and galactose, which are absorbed to the lumen of the intestine by the secondary active transport and all co-transporter, and from here the basolateral membrane, from the to the basolateral membrane, the GLUT2 are responsible to reach the basolateral membrane. Here the fructose absorption. Fructose start with F, so it is by facilitated diffusion, and F for fructose, F for facilitated diffusion, F for 4, that means GLUT4. I have already told that about the GLUT. Here the pentose sugar absorption of the pentose sugar, the absorption by simple diffusion. Next is the digestion of the protein. Digestion of protein start with stomach. Here the protein is break to peptides by pepsin, and pepsin is formed from the pepsinogen, which are in active form and secreted from the chief cell. And here for the activation of the pepsinogen, 2-pepsin requires HCl. This HCl break pepsinogen to pepsin, and pepsin digest protein to the peptides. Here most important thing in the intestine is that the trypsinogen, the secretion of the trypsinogen in the inactive form, in the gene, that means inactive form. These are converted to active form in form of trypsin by the presence of enterokinase or enteropeptidase, and also trypsin activate trypsinogen. Here the N-terminal peptide are removed. You have to remember N-terminal peptides are removed from the trypsinogen to becomes a trypsin, which are active form of the trypsinogen. And these are main enzyme for the activation of the chymotrypsinogen, proelastase and procarboxypeptidase. This converted to active form, respective active form. Here the protein is digested in the stomach by the pepsin to polypeptide, and after that the trypsin, chymotrypsin, carboxypeptidase, elastase, it converted to oligopeptide. Here the oligopeptide get converted or the break into the amino acid, simple amino acid, by aminopeptidase or dipeptidase. The endopeptidase or endoproteinase are the elastase, trypsin, chymotrypsin and pepsin. These are known as endopeptidase or endoproteinase due to have nature to break the middle or inner bond, peptide bond. The terminal peptide bond are broken by the carboxypeptidase and aminopeptidase. So these are called exopeptidase, and elastase, trypsinogen, chymotrypsin and pepsin are known as endopeptidase. Absorption of the amino acid by secondary active transport with sodium, with co-transporter like glucose absorption. Next is the digestion of the fat. Fat digestion starts from the mouth. Here the lingual lipase, Abner's gland start digestion of the fat from the mouth. Then after gastric lipase, and then after main digestion start in the duodenum is the pancreatic lipase. The first lipase will start acting after emulsification. Let us discuss the thing about the fat digestion. The bile salt which are present in the bile juice have property to emulsify the fat. The emulsification includes the triglyceride to a small fat droplet have size 1 millimeter. So emulsification requires the diameter of the triglyceride which converted to a small fat droplet. The size of a small fat droplet will be 1 millimeter. And for the absorption of the digested fat, it should have to form micelle. That means a small fat droplet to much smaller. That means for micelle formation, the size should have 2 to 5 nanometer, and after that their absorption takes place. The detergent action due to amphipathic nature and bile decrease surface tension. Here the inactivate pancreatic is also inactivate pancreatic lipase, and while that activated by colipase. For the fat digestion, first required to emulsification, then lipase acts on it. So first digestion of the fat require emulsification. When emulsified fat, then it form micelle, and the lipase acts when fatty food is emulsified. The emulsified fat, the action of the pancreatic lipase, this convert to free fatty acid and 2-monoglyceride. Here the structure of that. Okay, here 2-monoglyceride and 1 free fatty acid. The absorption, the triglyceride, lipase, colipase, bile salt, it convert triglyceride, that means more than 10 carbon. The criteria that more than 10 carbon get breakdown to the free fatty acid and 2-monoglyceride. Then free fatty acid are absorbed to the parietal cell. After absorption, this cell, intestinal cell, get absorbed and it combined again. Here the requirement is that if the fat have more than 10 carbon, it have to break to simple form free fatty acid and 2-monoglyceride, and then after in the cell it get again combined and becomes triglyceride, and this triglyceride get converted to chylomicron, and this chylomicron is transported to the lymphatics. Next is the medium chain. What will be the medium chain triglyceride? Medium chain triglyceride, that means which have less than 10 carbon and are not require bile salt and lipase. So you have to remember this, all the digestion process for the fat. The property is that triglycerides should have more than 10 carbon atom. Less than 10 carbon, not require emulsification, not require bile salt, not require lipase. It directly get absorbed, and short chain fatty acid produced and absorbed in the colon like acetate, propionate, bitrate. The next is the substance absorbed mainly in the duodenum. This is also important. So you have to remember that in the duodenum, the calcium and iron are mainly absorbed. DCI. The substance absorbed mainly in the jejunum. Jejunum is the main for the absorption of the sugar, amino acid, fat, vitamin, both water soluble and insoluble. But B12 is less absorbed in the jejunum. And most of the water are absorbed in the jejunum, and electrolyte like sodium and the chloride. Next is the substance absorbed mainly in the ileum as the bile salt and B12. So B12, maximum absorption of the B12, bile and bile salt takes place in the ileum. Thanks for watching my video, and please subscribe my channel, watch my channel till last. All videos are most important for you. And any comments, please write in my comments box. This is the part first, and part second I will upload the video of the GIT motility related. So thanks again for watching my video. Have a nice day.